Hey guys, welcome to AP Biology, and this is for those of you who are not in class today. So let's go ahead and talk about cells, and I'm going to focus on the parts that Mr. Anderson did not talk about. So we'll jump right in. First of all, cells are small. Okay, they're real small, and so you need a microscope to see them. We'll be doing a microscope lab next week. But there are basically two types of, well, there's lots of different types of microscopes, but the one that you are most familiar with is a light microscope. And it basically is able to look at cells that are living, but it can't look at very small cells. Or actually can look at very small cells. We just can't see a lot of the details. We can see the nucleus and maybe some of the mitochondria and things of that nature, but the things like ribosomes or the cell membrane, that is, we need a special type of microscope called an electron microscope to do that. Okay, so again, with a light microscope, you're going to see down to about 2 microns, which is about the size of a small bacterium. It's right in this region right here. And as you can see, smallest bacteria, viruses, and ribosomes, and proteins and lipids, those are not available with a light microscope, mainly because of the physical nature of light. It's just not that small compared to electrons, which are really, really, really tiny. And that's where we use electron microscopes. And there's basically 2 types. We have a transmission electron microscope, which is great for, as I say up here, looking at the internal structure of a cell. Okay, and then there's a scanning electron microscope, and you can look up some of the images of these online, and they're really beautiful, especially of things like insects and things of that nature. But it almost looks three-dimensional. Some of the drawbacks with using electron microscopes, however, is that you cannot look at living organisms. They have to be dead because you coat them with a thin layer of gold. With light microscopy, we can actually look at living organisms, which we're going to do next week. All right, so some of you guys may wonder, well, how do biologists know all this stuff? How do they know how ribosomes function? How do they know that a Golgi apparatus can do this or an endoplasmic reticulum? Well, we do that by kind of breaking the cell apart and looking at the individual parts in vitro. Remember, in vitro means not in a living organism. In vivo means in a living organism. So we can take out the ribosomes, we can put them into a test tube, we can add messenger RNA, and we can add various types of amino acids, and we can watch how the ribosomes actually take those amino acids and the messenger RNA and then build a protein on that. And we can design our own proteins that way. And so that's how we basically know how these various organelles work. And sometimes we can also look at what happens if we remove an organelle from a cell and see how that affects the organism too. Okay, and the way we actually do that is by basically cell fractionation. Basically, we break the cell up with a blender, we put it into a centrifuge, spin it. The very light stuff like ribosomes will float to the top, and the very heavier stuff will be down in the bottom in the area we call the pellet. The area above the pellet is the supernatant. We will be doing this technique in some of our future labs, so I want you to know those terms, and that's all I want you to know about that. Okay, so this is an important slide discussing the cell theory. So there's basically 3 major parts to the cell theory. 1, all organisms are composed of 1 or more cells. And I do want you to know that some prokaryotes will form aggregations with other prokaryotic organisms, not of the same species, in something we call a biofilm. You have a biofilm in your mouth, and that's plaque. Okay, different species of bacteria and other things all living together, doing things together that they cannot do alone. So one bacteria might be able to metabolize one type of sugar, the others can't, but they are able to use the waste products of the other bacteria in order to gain more energy and things of that nature. Biofilms are actually very interesting, and there's some really cool research projects going on about the use of biofilms to do things like clean up oil spills or toxic waste and things of that nature. There are single-celled eukaryotic organisms. Most of the protista, which are eukaryotic organisms, are single-celled, and there are multi-celled bacteria. So I need to update that slide, put it that way. Anyways, cells are the smallest unit of life. That's where life begins as we know it. Okay, so at the cell, an organelle is by its own is not living like a ribosome or a mitochondria. Um, Mitochondria maybe we'll talk about that a little bit later, but so the cell as we know it, however, is the basic unit of life. And the other thing, part of the cell theory, is that all cells came from pre-existing cells, except very early in life's history, when we basically had abiogenesis, the formation or the creation and formation of life, the evolution of life through abiotic means. Okay, so we get into that a little bit in evolution, but it's kind of a separate topic. All right, so one of the things I do want you to know that life has been on this planet for the latest number I saw was almost 4 billion years. We know the planet is about 4.6 billion years, and those very early life forms were most likely heterotrophic bacteria. Heterotrophic organisms are organisms that have to eat something else. They cannot make their own energy like a autotrophic bacterium. An autotrophic bacterium can use either chemical energy or sun or light energy in order to make the sugars that it needs to survive, whereas heterotrophic bacteria need to consume stuff that is in the environment. And we do know that photosynthesis was a much later evolutionary product, probably somewhere in the neighborhood of 2.7, 2.8 billion years ago. We believe photosynthesis, I should say, we have evidence that photosynthesis evolved about that time point, mainly because of the chemical nature that we see in rocks of that age. Okay, we see evidence of oxygen, and oxygen was not really abundant at all in the atmosphere or in the ocean water up until the evolution of photosynthesis. But what we're looking at here in this picture are stromatolites. Now, stromatolites, it looks like a rock, but what it actually is a combination of biotic in the form of cyanobacteria, and cyanobacteria are able to photosynthesize. And sand. And these are found in Australia in warm, saline, very salty waters, okay, that are relatively not hit by a lot of waves and things of that nature. So on top of this rock-like formation is a very, very, very thin layer of living bacteria. And as the tide goes in and out, that bacteria, here it is at low tide, sometimes gets covered up with sand that's in the water. It just kind of settles in on top of that. And then bacteria grow and above that sand, and then more sand comes on and then gets covered up, and then more bacteria grow on top. So if you look at some of these fossil stromatolites, what you see are if you cut the rock in half, you see these nice banding patterns, and the bands represent the living bacteria. Well, they're dead now because they're fossils, and this rock is about 3 billion years old. So this is some of the evidence that shows us that photosynthesis probably evolved somewhere, again, between about 2.7 to 3 billion years ago. I'm not exactly sure that exact date, but it's somewhere in that region. Okay, so they've been around for a really long time. Life has been a long time on this planet. We don't see our first eukaryotic organisms, multicellular organisms, until about 1 billion years ago, maybe 1.5 billion years ago. So most of the life's history on this planet were basically bacteria, and that's it. No life on land. All the life was in the water. Okay, on this particular slide, just kind of looking at the general size of both prokaryotic, eukaryotic, and viruses. Okay, so the prokaryotic cell, you can see the bacterium there is much smaller, somewhere in the order of 10 to 1,000 times smaller than a typical animal cell, and a virus is even smaller than that. And this particular virus is a T4 bacteriophage. It's a virus that infects only bacteria. And if you've ever been swimming in the ocean and got some water in your mouth, you probably got hundreds of thousands, if not millions, of these bacteriophages in your mouth. They're very common in ocean water. And viruses are common everywhere. Your body's loaded with them. They're just everywhere. We did on our cells, on the ideal cell size lab, we started going through some of these calculations. I expect that you are able to calculate the surface area of a cube. We'll practice with how to do surface areas of spheres in class, but I expect you to be able to do that. And I also expect you to know that it's the surface area to volume ratio that's important. And if you look at the first cube, which has a length of 1, it has a total surface area of 6. 1 times 1 is the surface area of one side times 6 sides. And so it has a surface area of 6. Its volume is 1 times 1 times 1, or 1. And therefore, its surface area to volume ratio are 6 units of surface area to 1 unit of volume. Okay? Whereas on the cube with a side of length of 5 has more surface area, 150, has a bigger volume, 125. But when you divide the surface area by the volume, you get a 1.2 units of surface area per 1 unit of volume. So there's a lot less surface area servicing that interior part of that cell. However, if you take that large cell and you now and you now divide it up into smaller 1-unit cells, you can see that the surface area increases dramatically, 750. Volume is also up, but the surface area to volume ratio remains the same as it was with the 1-unit cell at 6. So, and again, why is that important? Well, if you think about it, the surface area of the cell is basically what allows the cell to import nutrients into it and export waste out of it. Things have to diffuse through the cell membrane. The further away they are from the cell membrane, such as in this large cell, the longer it's going to take for it to get out. And there's just not as much area for material to move in and out of compared to the small cell. So a large cell, a cell that was too big with too small of a surface area to volume ratio, would either die of starvation or drown in its own waste, which sounds absolutely disgusting. So that's the reason why surface area to volume ratio is so important. I'm just going to show you a couple of pictures of a typical animal or typical cells of both plant and animal. And just as you can see, this is very packed. You would never, ever, ever see a cell like this in nature. This is just a cartoon showing you some of those major cell organelles. I do want you to know how closely associated the endoplasmic reticulum is with the nucleus and also with the Golgi apparatus. And that's going to play a role when we talk about protein transport, protein movement, and things of that nature. Okay. Also notice down here things that you do not find in animal cells: chloroplast, central vacuole, and tonoplast, a cell wall, or plasmodesmata, which are little holes in the cell wall that allow for communication between cells and between plant cells. Okay, Mr. Anderson, I think, did a good job on this. This is a plant cell. We see the cell wall around there. We see the plasmodesmata, the little holes that allow for the movement of water and chemical messages and sugars and things like that into and out of that particular cell. Some of the things we do not see in plant cells are lysosomes, centrioles, and flagella, but some of the plants like ferns and mosses do produce sperm cells that have flagella and do swim. All right, so how did all those organelles develop? And we have the endosymbiotic hypothesis right here. I'm trying to figure out where to put me. I can't figure it out. I'll stick it right there. Okay, and basically what the endosymbiotic hypothesis says is that organelles such as the chloroplast and the mitochondria were once free-living bacteria that became engulfed into a proeukaryotic cell. We believe this happened sometime about 2 billion years ago. So basically, how do we know that? Where's the evidence for that? Well, if you look at either both chloroplasts and mitochondria, they have a lot in common with bacteria. A, they're about the same size. Okay. B, they have their own DNA. And not only do they have their own DNA, but their DNA is in the same form as a prokaryotic cell. Our DNA as a eukaryotic organism consists of multiple linear chromosomes. In humans, we got 46. Bacterial cells, however, prokaryotes, only have one chromosome, and it's in the form of a circle, although it's folded up and everything like that. But if you stretch it all out, it would be a single circular chromosome. Okay, and that is what chloroplasts and mitochondria have. Another piece of evidence is that both chloroplasts and mitochondria have a double membrane system. They have one membrane, which was their original membrane, and then they have an additional membrane on the outside, which is a result of them becoming engulfed and put into a vacuole in the proeukaryotic cell. Okay, so that's how, and we have lots of it. There's other pieces of evidence, the type of proteins they make, that their ribosomes themselves also are very of chloroplasts and mitochondria are much more similar or almost identical to the ribosomes you see in bacterial cells, which are different than the ribosomes we see in eukaryotic cells. Excuse me. Jumped it over. So in a nutshell, here's basically what has happened. Let me move my picture back down. Doesn't want to move for some reason. We have the two independent bacteria. This is the proeukaryotic cell. One engulfs the other. It puts it into a little vacuole. So it puts some of its own membrane around it. And that's the reason why we don't see it as a bright purple one right here, because it's completely covered in another membrane. And that is the beginning of that double membrane system. And both bacteria benefit from this arrangement, and it makes this one more likely to survive and reproduce than one that doesn't have it. And so if it's more likely to survive and reproduce, it produces more offspring, so to speak. And those very quickly become the dominant form of bacteria in that particular system. It's just natural selection and evolution. There's really not much too complicated about it, but we'll go into a deeper dive on that in our Unit 7. Okay? Other organelles, what's going on? Other organelles, such as the endoplasmic reticulum and the nucleus, we think may have evolved through the process of invagination of the cell membrane. So here we see some DNA, and we see the membrane beginning to kind of infold towards itself, and eventually it will cover or surround that nucleus. Here we have some membrane-bound ribosomes, and those ribosomes during that invagination will eventually become part of the endoplasmic reticulum. So that's two processes: endosymbiotic hypothesis, which we have lots of evidence for, and invagination for some of the other organelles, such as the nucleus, the endoplasmic reticulum, the Golgi apparatus, and so on. Okay. Having problems with advancing. All right, so we are going to go do a deep dive into the plasma membrane. We're not going to do that today, but we will do that starting on Friday when I am not here. Okay, I'm going to give you some other stuff for that. All righty. So do want to talk a little bit about prokaryotic and eukaryotic cells. They do differ, but they have some similar properties, and I expect that you know what these similar properties are. First of all, that they are surrounded by a plasma membrane, a phospholipid bilayer. Okay, they have a cytoplasm, also called a cytosol. So you should know those two words, cytosol and cytoplasm. They mean the same thing. They all contain DNA, in the form of chromosomes. We have multiple linear chromosomes. Prokaryotes have a single circular chromosome, but it's DNA. Okay. Also, all cells do have ribosomes. Okay, we need to be able to take the information in the DNA, convert it to RNA, and then have ribosomes convert that into proteins in that flow of information. So make sure you know what all those four commonalities of all cells are. Okay. All right, so bacteria, as I say up here, are relatively simple cells, and there's two basic types of bacteria. We have gram-positive and gram-negative, and that's basically referring to the structure of their cell wall. Bacteria do have cell walls. However, unlike plants, they are not made out of cellulose. Instead, they're made out of a sugar protein called peptidoglycan. Peptide, like the peptide bond, and glycus means sugar. Okay, so peptidoglycan means a protein sugar. Gram-positive bacteria, which are the ones that are really susceptible to antibiotics, we can get rid of them with antibiotics, have a cell membrane surrounded by a peptidoglycan layer. And these antibiotics work by basically preventing the peptidoglycan layer from forming, so the bacterial cells really can't divide. However, gram-negative bacteria have a cell membrane, a peptidoglycan layer, and then another cell membrane on the outside. And that prevents a lot of bacteria a lot of antibiotics from affecting that peptidoglycan layer. So gram-negative bacteria are a little bit nastier. They're harder to get rid of, and oftentimes they're more pathogenic. Oh, Lord. Okay. Yeah, there's that peptidoglycan, a protein saccharide layer. And let me move my picture once again down over to here. And the gram negatives, as I said, are more pathogenic. This is having problems today. So here's just some of the things we see with bacteria. There's no membrane-bound organelles in bacteria, so there's no endoplasmic reticulum, there's no Golgi apparatus, there's no vacuoles, there's no lysosomes, nothing of that nature. They do have a single circular chromosome. However, here you can see that is oftentimes very folded up very tightly because it's still a big chromosome because it's a tiny cell, and that's what we call the nucleoid region sometimes. Okay, some bacteria will have flagella, some have cilia. Actually, they don't have pili, they call them, not cilia, but pili. They have ribosomes, which are not membrane bound. They have a plasma membrane, and they have cytoplasm. And that's about it. Okay. Very different than a eukaryotic cell, remember, which has all those different organelles. And all those different organelles lead to a property we call compartmentalization. There's your 10-cent word for the day. And compartmentalization just basically means that in a eukaryotic cell, you can have multiple environments. Okay, we can separate different things. It's almost like the analogy I like to use is the school. Here at Trustful, we have a big school, but we have all of our classrooms are in different rooms, and we can teach biology, we can teach English, we can teach culinary arts, we can teach gym class, we can teach all sorts of things in this school because the rooms are all separate. Kind of like a eukaryotic cell, there's different environments in those rooms. But in a prokaryotic cell, it would be like a one-room schoolhouse. Yeah, you might be able to do it, but it would be really, really, really hard. It wouldn't be very efficient. So we're going to get deep into this when we talk about photosynthesis and cell respiration and the role that membranes play in separating and creating separate environments, which sets up what we call concentration gradients, which allows for the production of ATP and all sorts of things. And again, as I say up here, many enzymes are built into the phospholipid bilayer or into the cell, into the membrane of the different organelles. And that's where all the action happens, or on the membrane, as you will see in photosynthesis and cell respiration. And that's the lecture for today. I hope you enjoyed it, and we will see you soon.